Okay. Never know how to begin these things. All right, we are now to week 2, chapter 16. Week 2, we cover chapters 16, 17, 18, and 19. I tried to warn you at the beginning of Bio 124 that we cover a lot of material. And oh my gosh, do I mean a lot of material. Now, if you actually look at the book PowerPoints, compare them to my PowerPoints, you're going to see that I slimmed them down substantially. It's because, like I said at the beginning of the quarter, it is not possible for me to cover every last part. I can only facilitate, I help, I expand on some concepts, not all. So if you cover something in the textbook or in the book PowerPoint, you want more information about it and I didn't cover it in one of these lecture videos, then all you have to do is email me, ask me. Once we actually get to be in person, ask me in person. All of that works. So without further ado, here is chapter 16. Intro. Microorganisms residing in and on your body outnumber your own cells. That means the things that live in and on you, you have more of than your own personal cells. If I were talking with my son Pax, we would have a really fun philosophical talk about this one, but we're not. This is our bio class, so we're just going to move on. So what's going on with this, why it's important, is about how different things can mess up our microbial communities inside our body. So this can make us more susceptible for a disease, can predispose us to certain types of cancers, and can even contribute to certain types of conditions. I'm going to probably disagree with some of the stuff here, but we're still going to cover it. I do not recall anywhere in the textbook it actually saying how your microorganisms lead to autism, but we'll keep looking and see what they say. It does have something to do oftentimes with allergies, not all of them, but some, and irritable bowel syndrome and things like that. This is also the reason why many people, when they take antibiotics, end up with various types of yeast infections. So, big ideas. Prokaryotes and protists. That's pretty much what we're covering in chapter 16. Then we move up to a little bit more in chapter 17. If you remember, I told you Bio 124 begins with evolution. So we are starting at the bare basics, and then we're working our way up to people. Then we're going to talk more about how our insides work, not just people, animals and stuff like that, but mostly people. And I just thought of the eagle from the Muppets on that one, but mostly America. But anyway, we're going to be doing that, and then by the end of the quarter, we will talk about the big picture of the entire biosphere. So here we go. Bam. Prokaryotes. So these guys are diverse and widespread. They are very different, and they're all over the place. That's pretty much all I have to say. We talked previously in Bio 123 and last week about prokaryotes not having a nucleus and the eukaryote having a nucleus, because I said to remember EU means true. So that is a true nucleus. We are talking here prokaryotes. So these are before we evolved up to have the nucleus. So collective biomass of prokaryotes is at least 10 times that of all eukaryotes. This means we have more. Prokaryotes have an immense impact on the environment and on our health. And there are two very different types. We have Archaea and bacteria. I want to say it was the first PowerPoint of Bio 124, but I might have mentioned it just in the intro of Bio 123. But for the domains, Archaea is pretty much the catch-all. It's like if it doesn't fit anywhere else, that's where we put it. All right, here we go. We are starting right off with a checkpoint question. The number of bacterial cells that live in and on our body are greater than the number of eukaryotic cells that make up the body. Why aren't we aware of these trillions of cells? So why do we not even know that we have all these? Well, because we can't sense each individual cell, you might feel your finger, but you don't feel every single cell that makes up that finger. Kind of like that. Bacterial cells are even smaller. So if you're not noticing your own individual cells, why in the world would you notice all the little bacterial cells? And these guys coexist with us. You only tend to notice them when something has gone wrong. Like I said, if you've taken antibiotics and it's killed off your good biota that keep things in check, you notice the other stuff going crazy. So, determining the cell shape of microscopic examination is an important step in identifying. So basically, you're going to notice they have different shapes. The different shapes will dictate pretty much what we're going to call them because it explains what they do. So we have the spherical ones, the ones that are kind of round. These are the cocci. Then we have the rod-shaped, which are bacilli, and then these spiral ones. So we have two different types. We have the short and rigid one, which are spirilla, and the longer, more flexible ones. So these are all teeny tiny little microscopic suckers. And this is amplified, magnified, so you can see them. Now, almost all of them have a cell wall similar to a plant cell. Plant cells have cell walls, animal cells do not. So this, I'm telling you this because when we're looking at that phylogeny tree, it comes into play. Prokaryotes can be classified by shape and by reaction to a gram stain. If we had a lab where we could do this, we would totally be doing this. And it's where you stain it, and so you can see which one's which. I think I just lost where my little thing is. There we go. Okay, gram positive. I'm going to tell you, I just remembered purple positive because when we did this in lab when I was in school, I knew that the gram positive turned purple, and so that's how I could remember it. Because what it does is it reacts with the peptidoglycan. The walls of the gram negative bacteria don't stain the same. So here is I don't know what word was about to come out of my mouth. Here's an image of different bacteria. The purplish, purple question mark, they look more black to me, but the purple, those are the gram positive. The lighter colored ones are the gram negative. And then this is just showing you a bacterium that shows both the flagella and the fimbriae. Flagella, we talked about this previously in definitely 1, 2, 3, and I think at the very beginning of last week, but anyway, the flagella is like a long whip-like structure, and if you remember, it can go like this or it can go like this. It helps it move along. Checkpoint question: how can a microscope help you distinguish the cocci that cause staph infections from those that cause strep throat? Well, it's literally how they match up. Staphylococcus, that's how you know that it's a cocci. That's how you know that's the little round bacteria. Whenever you see the name and it ends with coccus, C-O-C-C-U-S, that means it's going to be those little round guys. So the staph is clusters and the strep are chains. Okay, populations of prokaryotes can adapt rapidly to changes in the environment. Oh my gosh, their generations are pretty friggin' short, so these guys can make changes really fast. Some prokaryotes form endosperms that remain dormant through harsh conditions. And yes, when I say a phrase like that, it means we're going to come to that word again at some point, and it's probably a good idea to understand what they mean. So, why does rapid reproduction produce high genetic variation in populations of prokaryotes? Well, because each time the DNA replicates, spontaneous mutations may occur. Cover this in genetics, and we cover this at the very beginning of Bio 124. Every next generation is better suited for that environment. So all the little changes that take place get passed on, and so it just keeps trying to fit, match, whatever. Prokaryotes exhibit much more nutritional diversity than eukaryotes. This is what allows them to inhabit almost every nook and cranny on Earth. You have this image in your textbook, figure 16.4, and it's trying to show you that you either use sunlight for food or chemicals for food. So if it's using sunlight, it's going to say photo. So we have photoautotrophs, which means it makes its own food, or photoheterotrophs, which means it's using it to bring in its stuff, mixing it with other stuff. Why am I using the word stuff so much? Photoautotrophs, like plants, are using the carbon dioxide and the sunlight to make their food. Photoheterotrophs have to pull in other compounds, other chemicals in order to mix with the sunlight. Then we have our chemical stuff. We have the chemoautotrophs. This is where the chemical they absorb mixes with the carbon dioxide and they make their own food. Or they take these chemicals and other organic compounds in order to make their food. So those would be your chemoheterotrophs. This is breaking it down. Again, just letting you know, showing you energy source and the carbon source because you have to take in the carbon. So which term would describe your mode of nutrition, as in yours, you as a human being? Chemoheterotrophy. The reason is chemo means compounds. So you have to take in compounds. Hetero literally means different. We've covered that quite a few times now. And trophy means pertaining to food or nourishment. So if you see autotroph, it means self-food or self-nutrient. If you see photoautotroph, photo means light, auto means self. Photoheterotroph would mean light, different, nourishment. So it means it's taking in the light, has to pull in carbon from somewhere else, and that's how it gets its food. Prokaryotes attach to surfaces and form highly organized biofilm communities that are difficult to eradicate, causing both medical and environmental problems. Before I cover the checkpoint question on why biofilms are difficult to eradicate, I'm going to tell you a little bit more about biofilms. The textbook is going to show you some different toxic ones and like where it fills up a pipe and things like that. I normally use one that everybody is aware of, and it's the plaque. It's right here. It's on your teeth. I don't mean the tartar that they're chipping away at. I'm talking just the before it gets to the tartar. The tartar's like hardened up. But if you have ever gotten a new mouthwash, you've scrubbed your mouth, you've brushed your teeth, you use this brand new mouthwash, and then later you're like, and you're just doing this with your finger and you're pulling it out and you've got that weird, gross, stringy stuff and you think there's something wrong, that means the mouthwash is actually working well and it's helping remove the built-up biofilm in your mouth. It's just kind of shedding, for lack of a better phrase. It's pulling all that old out. So when you do that and come up with that whole thing, that is literally biofilm that you're pulling out of your mouth. I just think that's an interesting thing to know. But why are they difficult to eradicate? Well, because it sticks to the surface it resides on. So again, plaque, all this stuff up here, it just sticks. And then they also stick to each other. And the outer layer of cells might prevent things from penetrating to the interior. That's why it's so much harder to get rid of. And since I keep talking about plaque, I'm going to go ahead and throw out there it's extremely important to get your teeth checked and for proper dental hygiene, because we will talk about teeth and how your dental hygiene affects your personal health a little bit later in this quarter. But just know, take care of your teeth. All right, bioremediation, it's all in the word here. It's the use of the organisms to remove pollutants from soil, air, or water. Prokaryotes are often used for this, including in sewage treatment facilities. This means it helps break, well, crap down. We'll just put it that way. It helps munch it and distribute it. The textbook has lovely photographs of the water treatment facility. I chose not to bother putting it in the PowerPoint. So, checkpoint. How might an influx of toxic chemicals affect the ability of a wastewater treatment plant to treat sewage? Well, if the toxic chemicals kill the prokaryotic decomposers, the organic matter in the sewage will not be decomposed. So if you basically kill off the decomposers, how do you expect it to decompose? Decomposers are here for a purpose. It's helping break the stuff down in a natural way to keep up the nice little cycle of life. So this is similar to when you take the antibiotic. The antibiotic does not only kill the bacteria that is messing you up, it's killing all the good bacteria in your body that we mentioned a little while ago. And then that means other stuff can proliferate. It can just go nuts because the guards are not keeping them down, basically. So same kind of thing here. I totally keep this one on purpose, because I talk about this in my environmental science class, and it is something that drives me nuts. This is during the Deepwater Horizon oil spill, Gulf of Mexico, and BP spread Corexic all over the place, and it's supposed to be a dispersant is what they say it is. And in reality, what it does is it just kind of gloops around a bit of the oil and drops it to the bottom of the ocean. So it doesn't actually make anything go away. It just hides it from view, and actually ends up being a lot more detrimental because you can't exactly clean it up once it's down there, and then other animals can come up across it, and it causes a lot of damage. So not a good thing. Bacteria and Archaea are the two main branches of prokaryotic evolution. Here we get to the evolution part. So, first major split in the history of life was the divergence of bacteria, and then later that separated two domains of Archaea and Eukarya. We're about to see pictures and stuff. Well, I don't mean of the actual evolution, but of a tree. Some of the main differences among the three domains involve the cellular machinery for gene expression. So, what characteristic is shared by bacteria and Archaea, but not eukaryotes? What's one of the big things I told you to remember about what eukaryote means? Right there. Prokaryotic cells do not have a nuclei or other membrane-enclosed organelles. So, nifty little table here for you to look at the bacteria, the Archaea, and the Eukarya. I think this textbook just really loves to use the red panda for a lot of things, but I'm not going to argue because he is insanely cute. So this is just a quick little breakdown on some of the stuff. Yeah, I'm not really going to ask you about most of this, just so you know. All right, you do want to know what the domains are, because that's part of your course objectives, is to know the three domains. These I like to talk about because we will cover some of these along the way. So we have the extreme halophiles, extreme meaning like way off on the other end. Phile, hopefully you already know, is love or lover, because if you remember from Bio 123 when we talked about hydrophilic and hydrophobic, hydrophilic was water loving. So here, halophile is salt loving. So these are the Archaea that are present in extreme salt conditions. Then we have the extreme thermophiles, thermo for heat. So these are the ones that love heat. And so you will find these near things like hydrothermal vents and whatnot. And then we have, I can never say these right, methanogens. There we go, that sounds good, right? These live in anaerobic conditions. And if you remember from Bio 123, how many times am I going to say that this quarter? I have no idea. I would love for somebody to count and tell me, because that would be entertaining. Anyway, anaerobic conditions. If you remember, I said aerobic meant it had oxygen. Anaerobic was no oxygen. So these are the ones that thrive in areas that do not have oxygen. So some Archaea are referred to as extremophiles. Why? Other than what I just said. Like literally, it's what we just talked about. I think it's kind of silly to put a checkbook question right there. Anyway, because they can thrive in extreme environments that are too hot, too salty, or too acidic for other organisms. And my cats are starting to argue with each other right over there. So you might get to hear that for a little while. Domain bacteria currently organized into 5 major groups. much I will leave. The cats have now been separated. And now Fang looks all kinds of confused. And Osiris will probably be hitting at the door in just a moment. But whatever. So, at least we took care of that. Alrighty. Domain bacteria currently organized into 5 major groups. It's these 5. Am I going to read them all off? No. Why not? Because you can read. It's just the main ones. There you go. Checkpoint question. How are, seriously, I cannot say that word, similar to the cyanobacteria? I can totally say cyanobacteria. They're both photoautotrophic. This you're not going to know unless you're looking in your book and you're looking at the picture, like straight up. And no, I'm never going to ask you this. Why did I leave this checkpoint question in the PowerPoint? I don't know. My apologies. So, pathogenic bacteria often cause disease by producing either exotoxins or endotoxins. If I haven't drilled it into your head in Bio 1, 2, 3, I swear I want a little counter that just goes bing in the corner. And if I was way more creative, I would totally add that in editing. I really, really would. But then I would have to watch my entire presentation, and I don't know if that's going to happen. But if all of a sudden you see a timer pop up, that'll be why. So exo is for outside, endo is inside. So exotoxins are bacterial cells secreted into their environment. So they're like secreting it off of themselves, so out. Endotoxins, lipid components of the outer membrane of gram-negative bacteria that are released when the cell dies or is digested. Digested, so that would be going in, so endotoxins. Now, certain bacteria, like some that cause anthrax and bacterial toxins, such as botulinum, I can't get the word right, and you'll understand why in just a second, other than the fact that I suck at words sometimes. These can be used as biological weapons. Clearly, you know anthrax is horrible. It's a bad, bad, bad mojo. However, a lot of people will also talk about, Botox. Why people do this, I don't know. But Botox injections is they're literally putting this toxin into your skin, and it is paralyzing the nerves, muscles, and whatnot in that general area. This is why the wrinkles get smoothed out. Because, you know, normally if you do this, wrinkles up, you do that, and yeah, there we go. It wrinkles. Everything you do, you see, I have like little wrinkles there when I try it. If you get Botox injections, have you ever seen somebody, their eyebrows still kind of move and it just looks perfectly flat? And it's because they have killed or deadened these for a period of time until they heal enough, and then the people go get more Botox. Ew. And the thing is, this was discovered accidentally, and I think it's hilarious that it was discovered accidentally, because how do you accidentally inject a toxin into somebody's face? Just gonna throw that out there. It's really disturbing. And yes, I know many people wish I would hurry and just change the slide, but why? Why do people do this? Okay, so contrast exotoxins with endotoxins. Well, we just talked about them. Exotoxins secreted, endotoxins, outer membranes of the bacteria. There you go. Now, this one, they've changed it up since the last textbook. These two people, they found that the peptic ulcers were usually caused by this particular bacterium. Now, the word usually is a very, very, very science. The more you read science-related stuff, the more you will notice it's a rare thing to see is, does. You normally see things like normally, usually, primarily. Things like that. I just think it's funny. I like it. But anyway, they found that this one particular bacterium tended to cause ulcers. And how they proved this was the guy actually ingested it to give himself the ulcer, so then he could take the antibiotic to kill it in order to prove, hey, look, that's what it is. There were so many misunderstandings about ulcers at the time, and so this was kind of cool. So we have ghrelin, which is a hormone. It's known as the hunger hormone because what happens is it stimulates the appetite, then it makes you eat, and then that promotes the fat storage. So when your stomach is empty, this output goes up and it's like telling your body, hey, we need the food, we need the food. You eat the food, your stomach is full, and so it stops kicking this out, telling you that it's hungry. And that's literally how it works. And so if you stay up late at night or you're up super early in the morning and you see the really dumb infomercials and they're talking about how we can trick this hormone and we can do that, this is one of the ones they actually talk about. So according to a study published in 2012, infants treated with antibiotics before the age of 6 months were more likely to be overweight at the age of 3. So would this result support the hypothesis that an absence of the H. pylori is a factor in causing obesity? What do you think? Possibly. Because they said that that was a bacteria, and so the antibiotic would kill it off. That's the microbial community. Now, they did not look only at H. pylori. They looked at, just a grouping. But it pretty much is about the same thing. If before the age of 6 months you're killing off the natural biota in the body, it's going to be harder to let it, I want to use the phrase symbiotic, but that's not quite what I'm going for. But it's going to affect how the body balances. What is happening is extremely important in those developing times. Now we are to protists. These are extremely diverse assortment of eukaryotes. So the protists are mostly unicellular eukaryotes found in a variety of aquatic and moist habitats. These might be autotrophic, so they make their own food, and these will be algae. Then we have heterotrophic, which are normally the protozoans. And then we have mixotrophic. Mixotrophic is pretty much what it sounds like. It's a mix between the two. Sometimes they use photosynthesis and sometimes they take in other organic compounds in order to make their food. So they're both. So what is a general definition for protist? This is a eukaryote that is not an animal, fungus, or plant. So again, catch-all. This guy falls in there and it's just a bunch of weird things. They're like, well, this is not an animal, this is not a fungus, this is not a plant, but it is eukaryote, so it's going to plop over here. So, fossil fuels, organic remains of organisms that lived hundreds of millions of years ago. And yes, hundreds of millions. This is why we say that it is a non-renewable resource, where it may have come from actual living stuff that is now rotted to the degree that it is a fossil fuel. Humanity is not going to be here in the amount of time it would take to make more. So there you go. Diatoms are thought to be the main source of oil, and I mention this because I really hope you look up some pictures of diatoms. They are really, really pretty. They look like teeny tiny little bits of glass. I know somebody who studied them. Dr. Ninow, he was at Valdosta State University, and he was actually studying them in the Gulf of Mexico after the BP oil spill. And so he was showing how the leak was still continuing. There was still damage years later, even though people had stopped talking about it. And then coal was formed from primitive plants, and it's just from the pressure for that amount of time. Researchers are working on methods of growing diatoms and other algae as a source of biofuels. This way we can get away from our love affair of fossil fuels, which is killing us. So, checkpoint question. What characteristics of unicellular algae make them attractive candidates for the production of biofuels? Well, you can reproduce them. We don't have to wait hundreds of millions of years. And this would not occupy farmland that we need for growing food crops. This means we could have our oil, it wouldn't be oil, but we could have our fuel and we could still actually, have fields of produce for people to eat. So we could have both. Oh, and then I just thought of that old El Paso commercial. Why not the blue balls? Anyway, here we go. Second supergroup in our survey of protists refers to an excavated feeding groove possessed by some members of the group. So here we have some excavates that are anaerobic protists that have modified mitochondria, and these include the parasite Giardia and then these other couple. I deleted the slides that showed all the pictures of these. Figured we'll just talk briefly about them. Giardia, best way I can describe it, bubble guts. This is like if you're traveling somewhere and they say don't drink the water. Certain fresh water areas like wells and stuff like that tend to have this living in there, and then people drink it and then they're like, if you've ever heard of the phrase runner's diarrhea, that's one of them. Yeah. The other one I want to mention is the termite endosymbionts. Termites, I find this fascinating. Termites cannot actually digest what they're eating. I mean, most of us can't. There's things in our gut that help us break it down. But the termites, why I find this one fascinating, is the main way that exterminators get rid of termites is they put the stuff out that actually kills the little suckers living in the gut of the termites that can digest everything. I'm looking down because I swear I'm expecting my cat to jump up on the table now that he doesn't have someone to fight with. Anyway, when they kill off the things inside the termites' guts, the termites keep eating, but they can't process anything, and then they just... And years ago when I was teaching a lab where we had the whole setup and we had this microscope at the front of the lab that I could hook up to the projector so I could project what's up there. We had this lab where I would take one termite and I would squish it. And there's the cat. Oh, there you go. And I would squish the termite so you could see what was in its gut. And yeah, it's mean, but it was really cool to see all the stuff. And yep, he made me move this from where I need it. And there we go. Whoops, nope, that's the wrong one. Don't want to end the show. So, I really need to just get a clicker, a real clicker. Okay, then we have this supergroup joins two well-established clades. We have basically, these are the amoebas, and we have fungi. Amoebas have what's known as a pseudopod. That means false foot. Pseudo means false, pod means foot. And so they like throw out this big fake foot. And the coolest thing about amoebas is they're actually predators. So they will throw out this fake foot and then engulf their prey and just pull it in and absorb it. And I realize I do think this is the PowerPoint I normally show. I normally show this really cool thing about bioluminescent dinoflagellates with an enzyme of luciferin. And I need to find that and show you because it's really cool. Okay, then we have plasmodial slime molds and cellular slime molds, and then we have fungi and animals. So, checkpoint question. Contrast the plasmodium of a plasmodial slime mold with the slug-like stage of a cellular slime mold. We're not really covering slime molds in our class. However, if we were, plasmodium is not multicellular, but it's one. A slug-like stage of a cellular slime mold consists of many cells. So, not really covering that one. And then we have this supergroup, which contains red algae, which are mostly multicellular and include species that contribute to the structure of coral reefs and species that are commercially valuable. This is basically slowly climbing up the evolutionary ladder until we get to plants, which we're getting to before too long. We have alternation of generations. We have haploid gametophytes and diploid sporophytes. Haploid would be that lowercase n, diploid is the 2n. We covered this when we were talking about mitosis and meiosis in Bio 1, 2, 3. I wanted so badly to say it with me. I really did. Yeah. Okay. This I kept in here. I had no reason to. It's the Volvox. It's just one of my all-time favorites. And yes, I do have a favorite algae. I know it sounds really stupid, but I used to get excited whenever we had a live sample of this and I could look at it in the microscope, because these guys are actually, they all live together in like a little colony. And so what looks like one giant just sphere, it's a whole bunch of little ones that are working together, and they look like these tiny little really cool marbles that are rolling around. If you want to look it up on YouTube, see if you can find any so you can see it under a microscope, that would be really cool. I love these things. I don't know, just do. So, how does chromosome number differ in the gametophyte and the sporophyte in the alternation of generations life cycle? Well, there you go. The gametophyte is haploid, which is N, and the sporophyte is diploid, which is 2N. So pretty much the gametophyte is still going to be the same thing as like when we covered it before, egg and sperm. The egg is a haploid, the sperm is a haploid. When they merge, when they come together, then that's where we have our fertilization. Here it says fusion of gametes. That gives us our zygote. The zygote goes to the sporophyte, and that has part from mom, part from dad, and so that is the diploid. Oh, I think we're almost done. So, increased complexity often makes more variations possible. We know this. We covered this in genetics. The more complex, the more differences there can be, which means the more likely something's going to survive. I mean, that's part of why we have the whole biodiversity, right? Multicellular, blah, blah, blah. Wow, why can't I do that? Things with multiple cells, how's that? We have brown algae, we have fungi in animals, and then we have red and green algae in plants. That's how they break up. Now, here's the phylogenetic tree that I said a while ago that we were coming to soon. It's just breaking them down, saying that way back here we started with some ancestral eukaryotes and then it broke off. And notice green algae, way removed from where we are, and the fungi is more closely related to the animals than it is to the plants. And that's why you have to remember that a fungus, a mushroom, is not actually a plant. It's a totally different beast. This is just trying to bring it in a little closer so you can see detail. In what way do multicellular organisms differ fundamentally from unicellular ones? Well, unicellular, one. So all the functions of life are carried out within a single cell. Multi, literally more than one. Multicellular organisms have specialized cells that perform different functions. So unicellular, one, multicellular, many. So you have everything's done in one cell, or we have different cells that do different things. So finally, we get to the end. So you should now be able to do a whole bunch of these things. A lot of these you probably need to read the book and go more into. Told you 4 chapters in 1 week is a lot, so I have massively slimmed this down. And then that brings us to letting you know our next one is going to be basically the evolution of plants and fungus. We're going to learn about gymnosperms, angiosperms, and stuff like that. And I don't know, I just really like this picture. I think mushrooms are fascinating. I think they're really cool. And I am highly allergic, so I can't go play with them. And sometimes I wonder if when something hurts you, it makes you more fascinated by it, because how many people are fascinated by just like these huge creatures that, would eat you up? Or is that just me? I don't know. My kids say that I'm going to be killed by an animal one day because I just don't know better. That I'm basically Hagrid. I don't think I'm actually going to ask you a question for the lecture assignment in this PowerPoint. Probably going to ask it in the next 1 or 2 chapters. I'm trying to force you to have to actually watch all of these. So it's more like when you come to class and you have to at least sit in the classroom. just so you're warned. And yeah, I told you I'm never going to tell you which one it's in. And I've graded everything from the previous one so far, and just want to make sure you know, if you listen to this part, you're probably not one of the people I'm talking about. But if you are, and you looked at the feedback and you came back, yes, I do very specific questions because I need you to prove to me you listened, not that you just opened it up and picked a couple of random slides to comment on. So I do come up with very specific questions for your lecture assignment. That's it for this chapter, and we'll be doing the next one soon. Guess that's it. Peace out.